Thank you. I also want to thank Dr. Twilley and Turner for introducing this talk. Some of what they said will make it easier to follow along with what I want to talk about. I'm going to be talking today about some of the work we've been doing looking at the impacts or basically understanding the salt marsh biogeochemistry and microbial community dynamics following the oil spill. I'm the one giving this talk, but it is the result of the effort of a lot of people within our Coastal Waters Consortium. As Dr. Turner mentioned, we had 26 PIs the first go-around. 3 of us formed a biogeochemistry and microbial ecology subgroup, Ann Bernhard and Ann Giblin from the Northeast. As a result, a lot of the personnel was based in Louisiana out of my lab. We had a postdoc, John Martin, our first go-around, who was involved in a lot of the work that presented, and we have 2 new postdocs moving forward. But there have been a lot of people involved in this project. These are just the ones that have come through my lab. A series of RAs, graduate students, and undergrads, some of which have been funded as RU interns through Coastal Waters Consortium and some through NSF RU site program that I've been the director of. So this is the effort of a lot of people. So as has been alluded to in the earlier talks, these ecosystems are pretty complex. And if you're trying to understand the impacts of oil, which has got toxicity and physical impacts associated with it, there are a number of direct impacts of oil as well as indirect impacts that are mediated by the intermediary components of the food web that are impacted by oil. And this is what happens when you put a biogeochemist in charge of putting together a schematic diagram. It's sort of in the middle. Everything kind of flows through it. And so this is sort of the part that we're going to be talking about today. Our group is largely focused on the soil component here of the biogeochemistry and microbes, as well as interaction with plants. But if you want to understand the impacts of oil, you have to understand a lot more about the way these systems function. They're a lot more complex than that. There are a number of indirect influences on the oil effects. There are physical forcing, the hydrology and sediment delivery. If you do not have good baseline data, it's sometimes difficult to determine whether or not the difference you see between sites that received oil and didn't receive oil is due to the oil itself or something about the hydrology of that system that not only delivered the oil, but may deliver different sediment properties to that. So this is an important concept to keep in mind when you're trying to interpret your results. We also have a number of other stressors on these systems. We have climate change going on. We're seeing changes within their salinity regimes from both saltwater intrusion as well as management practices of trying to mitigate our land loss by introducing freshwater diversions. We have changes in inundation patterns that occur as a result of climate change, as well as this interannual variability. And we see changes in vegetation. Some of these are related to these changes in salinity and inundation. Some of these are climate-induced changes that associated with the expansion of black mangrove into these wetlands that were previously dominated by Spartina alterniflora. So you have to keep all these things in mind when you're trying to interpret what is happening at some component of your food web. Oh, and then on top of all that, we have an awful lot of pollution of nutrients that are coming down through the Mississippi River drainage system. So our group's overall objectives were twofold. First was to try to improve our understanding of temporal and spatial patterns in marsh biogeochemical process rates, the associated microbial communities responsible for carrying out those processes, and the factors that regulate these rates. It's important that we have a solid baseline. Despite the fact that our wetlands have been a really important part of the coastal environment in Louisiana for a very long time, we've done a lot of study. When we came to try to understand the baseline level, some of the processes we were talking about, we found we didn't have really good baseline data, particularly at the locations where we were trying to study. Once you understand something about the way these systems function, then you can start to evaluate the impact that oil exposure has on these processes in the microbial communities. So our group has largely focused our activities in the first 3-year funding cycle of Coastal Waters Consortium in these following areas, focusing on the carbon cycle, particularly looking at soil respiration and greenhouse gas fluxes, which also involves some other elemental cycles, plant production and decomposition dynamics. There's been a large emphasis on the nitrogen cycle, looking at nitrification, denitrification, dissimilatory nitrate reduction to ammonium, and anammox rates. Phosphorus cycle, looking at what phosphorus availability is like through looking at phosphorus sorption rates, looking at the iron cycle, particularly looking at iron reduction rates, and then looking at the relationships between these process rates, the communities, and the abiotic variables in oiled and unoiled marshes. Now today I'm going to focus on talking about a few of these different components and some of the research we've done related to those. These are largely trying to keep with the mandate of talking about our published results, or at least in most cases results that we have for manuscripts that are currently in review. There's a couple of points though that I couldn't resist showing you a little bit about. Hopefully that convinced you with the first set of publications about them. Okay, so just to talk about our rationale. Biogeochemical pathways are carried out by a number of different groups. It's important to sort of understand some of those fundamental differences. So you can think about it this way. We have sort of two main classes. We've got autotrophs, or self-nourishers, and heterotrophs, which are nourish others. The classic way we think about autotrophs, we think about photoautotrophs. Your plants, they get their energy from the sun and light. Well, there's another class called chemoautotrophs that get energy from reduced inorganic compounds. And then your heterotrophs, which get carbon and energy from reduced organic compounds. So that includes all of us. At least I don't know of any of you that may be phototaxic or anything, but I'm assuming most of you are heterotrophs. Okay. So when it comes to trying to anticipate, hypothesize about how we think an oil spill might impact these systems, our overall viewpoint was, before this study started, that chemoautotrophic pathways are often going to be more susceptible to pollutants than heterotrophic pathways, because they tend to be carried out by a more limited number of organisms under a relatively narrow set of environmental conditions. So an example here for the nitrogen cycle in soils, the lines that are indicated in red are ones that are carried out by chemoautotrophs and their chemoautotropic pathways. Those include nitrification and the process of anammox, which takes ammonium and nitrate and produces N2 gas. In contrast, two important heterotrophic pathways are denitrification, the conversion of nitrate to N2 gas, and dissimilatory nitrate reduction to ammonium, which is converting nitrate to ammonium. So as a result of this rationale, we hypothesized that ammonium oxidation, which is nitrification, methane oxidation, and anammox, if it was present, would be the most impacted in oiled sediments. And denitrification, dissimilatory nitrate reduction to ammonium, and methane production would be much less impacted. And that largely abiotic processes, like phosphorus sorption, which has to do with how sticky the components of the soil are for phosphorus, to use a real scientific term of sticky, will be the least impacted. Okay, so throughout the first part of the CWC, we basically were looking at three regions that Dr. Turner alluded to. We have some sites in Terrebonne Bay, in the western part of Barataria Bay near Grand Isle, and the eastern part of Barataria Bay near Port Sulphur and the Bay Jimmy region. In each of those regions, we had paired oiled and unoiled sites that were determined based on some of the oil analysis that Dr. Turner talked about. We were very concerned about the high erosion rates that we see in our soils. So instead of just going into the marsh and setting up random quadrats, we decided we wanted to sort of incorporate that and try to ensure that we had some plots that would be present in these marshes for a long period of time. So we set up looking at, for a lot of our studies, a gradient from the marsh edge, from 5 to 20 meters into the marsh. This resulted in, on the order of 50 total plots across the region, We have had some shifting of sites over time, and then we've added some reference sites near LUMCON and some sites along a salinity gradient over here in Barataria Bay. Our general sampling approach has been to try to take different approaches for asking different types of questions. We've utilized some high temporal resolution sampling. LUMCON is a really long way away from a lot of things, but it's really close to the marsh. That means that we can take advantage of the fact that these Terrebonne Bay sites are very close to LUMCON, and so we sampled them monthly through the growing season in 2012, bi-monthly in 2013, and then seasonally since that for biogeochemical process rates, microbial communities, soil and water characterization. We've also tried to take advantage of, whether it's directly in conjunction with it or very close in time, the sampling that Dr. Turner alluded to across the whole region, where they do it in sort of late spring and in fall, and then we've also tried to sample in July. So we've gone across the whole region on the order of 3 times a year, and we sampled a subset of biogeochemical rates across those and the microbial communities. And then we've done, set up a series of intensive samplings at a subset of those above sites and or controlled sites for some intensive field sampling or experiments. So we've done greenhouse gas responses to changing salinities, look at explicitly at the expansion of Abyssinia into Spartina marshes to see how that may change some of the biogeochemistry. Done some oil exposure. And then the big one moving forward is that one of the challenges of not having a baseline is not being able to sample these prior to the oil spill. So we're going to be building some marsh mesocosms at LUMCON that will hopefully be going into operation around the turn of the year. To show you what I mean by some of our sampling, this is our original Terrebonne Bay sites where we have these 2 pairs of oiled and unoiled sites. And then the pairs are separated by a larger distance than there is between the two sites that make up a pair. And then this is a sample from one of our oiled sites, an example of our gradient into the marsh. This gradient is really important for us because we have some sites, for example, this site up here, in which since we established our sites in May of 2012, have lost on the order of 10 meters of marsh. So we still have these 2 plots that we had at the beginning, but we don't have the other ones. So I told you I'm going to talk about a few different processes. One of those is actually a suite of processes that are associated with soil greenhouse gas fluxes. We sampled those monthly in 2012, bi-monthly in 2013, and 2014 on we've been doing seasonally. We're using the ventic static chamber method, except for when we have very high water levels, and we use the floating chamber method. And we calculate rates from changes in concentration from 5 time points during incubation. Okay, so there's a lot on this graph. We have carbon dioxide fluxes on the top, methane fluxes in the middle, nitrous oxide fluxes in the bottom. The cheat sheet version is over here on the left. This is over the entire year. The box and whisker plots for the unoiled sites on the left, and the oiled sites on the right. And then the right-hand panel is what those patterns look like over time, with the open bars again being the unoiled marshes and the gray being the oiled marshes. So our gas fluxes varied significantly with oil status. Oiled sites were lower in CO2, higher in methane, and lower in nitrous oxide, net fluxes from the soil to the atmosphere. We do see some variation over time within the season. In 2012, we had peak in CO2 and nitrous oxide that occurred early in the summer, with lower rates as we move towards the fall. We've seen no seasonal pattern in methane. So why do we see higher net methane fluxes from the soils in the oiled marshes? Is that due to an increase in methane production, or, as we hypothesized, a reduction in methane oxidation? The data that we have right now suggests that it may be due to changes in methane oxidizing bacteria communities. Their abundances tend to be higher in our unoiled sites than our oiled sites. This is seen throughout that 2012 year, in our Terrebonne marshes. And it's also been seen across the entire region. This is in July of 2012, which is consistent with the hypothesis that one of the responses to oil would be a suppression of methane oxidation. What else might be responsible for controlling greenhouse gas fluxes? We look at the soil properties. For net CO2 flux, we see that we have positive correlations with organic carbon, total nitrogen, and soil water content. But when we break it down and look at the unoiled, which are the open symbols, we see that that's where we have the strong relationship. We do not have a significant relationship at all for the oiled sites. Nitrous oxide, we do have significant relationships. They do not explain a lot of the variance for some of them, with the exception of total nitrogen. But interestingly, we do not see a difference between the unoiled and the oiled sites. For example, total nitrogen explains about 20 to 25% of the variance in net N2O flux in both the oiled and unoiled sites. So CO2 are positively related to soil organic carbon, nitrogen, and water. But it's only in the unoiled marshes. N2O fluxes are related to soil nitrogen, as well to some extent to the carbon and the C to N ratio. But it's similar between oiled and unoiled marshes. And methane fluxes are not significantly related to any of the soil properties. Water depth is an important influence on net greenhouse gas fluxes. Now, this is something that's not often looked at in our classic wetlands that show up in textbooks that are from the Atlantic coast that have a low marsh and a high marsh, because it's very easy to go out there during low tide and sample the marsh. When you're at sites that are of a tidal amplitude of 20 to 30 centimeters and your water level is driven by wind more often than it is by tide, you get to the marsh when you can get to the marsh. And as a result, we actually did a lot more sampling that span a large gradient of water inundation. And what you see here is that CO2 and methane fluxes were significantly higher when water depths were less than 10 centimeters for both unoiled and oiled sites. And N2O is significantly greater at lower depths when you combine both the oiled and unoiled plots. When your N went up, we could see a difference. So this is me going with something we do not have quite in review, because I'm waiting to see the rest of the story. But how long do these oil impacts persist? So this is what I showed you here in 2012. This is 2013 and 2014. You see, we still see significant differences between our unoiled and oiled sites in both of those years. So 4.5 years and counting. We've been sampling in 2015. Don't really have that data processed yet to be able to tell you about it, but we are seeing a consistent response in terms of the soil greenhouse gas fluxes 2 to 4 years post-oil spill. One of the things to think about when you're talking about greenhouse gas fluxes is the fact that they don't all have the same warming potential. We tend to think about carbon dioxide releases the atmosphere and how that correlates with climate change and warming, because that's the most abundant of the greenhouse gases. But in fact, methane has about 25 times the warming potential of CO2, and nitrous oxide is 298 times. So you don't have to have a very big flux of N2O to have a very significant impact that may be on par with what you see in some of the other gases. So if you convert all of these fluxes into carbon units, you can talk about sort of the radiative forcing due to the soil processes. And what you see here in 2012 is these CO2 equivalent fluxes broken down into CO2 in the gray bars, methane in the black, and N2O in the white. This is for each of the months of the year, and this is overall in the year. You see CO2 dominates the contribution to radiative forcing in Terrebonne Bay at a little over 80%. But when you break it down by oiled and unoiled sites, you see that in the oiled sites, the methane accounts for about 33% of that total radiative forcing, and only 4% in the unoiled sites. So we're seeing a huge shift in terms of the relative importance of methane as these sites become oiled. When you look at 2013 and 2014, you see that throughout the 3 years that methane accounts for 33 to over half of the radiative forcing from the oil marshes, and only 3 to 5% from the unoiled marshes. So methane is becoming an increasingly important greenhouse gas flux as these sites have been oiled. Okay, so now I want to switch gears and talk a little bit about the nitrogen cycle and talk about nitrification in general. First, so one of the first things that really struck us was how high our nitrification rates were. Louisiana salt marshes have the highest rates of nitrification potential in the literature. These are our 2012 rates that are in the Martin et al. 2015 paper, and our 2013 rates that are in a manuscript we're working on. Compared to other salt marsh sediments, which are carried out using the exact same techniques by my co-PIs, Ann Bernhard and Ann Giblin. So it's not that there's a bias in the way these are being done. They're being done by the same people. And in other estuarine sediments. However, despite what we hypothesized when we went across the region in 2012, we did not see a significant difference in nitrification potential in between our unoiled sites and open symbols and oiled plots in the black bars on any of the 3 regions. And we didn't see a big difference in the bacterial or the archaeal ammonium oxidizer abundances in these regions. Although the way it is depicted here, you do have a trend towards the unoiled being higher in abundance than the oil. So in 2012, we saw this seasonal pattern here. We had a peak in these rates in July. In 2013, we actually saw much, much higher rates earlier in the season, and July was actually not statistically different in the 2 years. So we have a different seasonal pattern. But more interestingly to me is that now 3 years post-spill, we actually are seeing a consistent pattern in Terrebonne Bay of higher rates in the unoiled marshes than in the oiled marshes. And early in the season, when these rates are highest, is when they're actually statistically significant differences. So there's something fundamentally different about 2012 and 2013. What is controlling these rates of nitrification? Having these really high rates, what's driving that? So if you look seasonally within Terrebonne Bay, we see organic carbon explains the highest percent of the variation, but it's only about 18%. But in the months that have the highest rates in June and July, they explain 35 to 40%. There are some other significant relationships with some other soil properties in these sites. We went across the region, we had a very similar pattern that organic carbon explains the majority of the variation. But interestingly, we saw a different pattern within each of the 3 regions. And all of them explain a lot more than they do overall, between 35 and 50% of the variance. But we do not see a significant relationship with oil, in the oiled sites with organic carbon, only in the unoiled sites. What about the microbial communities? If you look at the, what I've shown here is ammonium oxidizing bacteria. The ammonium oxidizing archaea show a very similar pattern. Nitrification does tend to increase with these abundances, but it only explains about 20% of the variance. And actually, it only explains it significantly within 2 of our 3 regions. But also surprisingly is that we actually see a stronger relationship with those abundances in the oiled sites than we do in the unoiled sites. And this is much less of the variation being explained by these abundances than Bernhard and Giblin had seen in the northeastern salt marshes. So we did some multivariate statistics to try to see if we could tease apart how these sites separate. And what we find is that we see stronger separation between sites than we do between oil status. So in the top panel here, we have Western Barataria in gray, Terrebonne in white, and then Eastern Barataria in black. And so we see a pretty strong distinction here. Western Barataria marshes are different than the other 2 regions. And for the most part, eastern Barataria are intermediate, but there is some overlap with terrebone sites. But we do not see a separation with oil status. And this first axis here has positive loadings with organic matter. So organic matter, organic carbon, and total nitrogen. And the y-axis, or axis 2, is related to increasing relative elevation and has positive loadings of nitrification potential, and then AOA and AOB abundances. So I want to go back to something that was very interesting to us, is that if you try to look at the correlations between the abundances of the microbes and the nitrification rates. What you see here are the R values for ammonium oxidizing bacteria and archaea within each of the 3 regions, terrebone, western Barataria, eastern Barataria, and then overall. Draw your attention to these boxes around the oiled, and we see that we have significant correlations only with oiled sites. None of these are with the unoiled. But in terrebone, it's only with ammonium oxidizing bacteria. Western Barataria, it's with ammonium oxidizing bacteria and archaea. And eastern Barataria, it's only with archaea. So we only have a relationship between what is thought to be the organisms responsible for carrying out nitrification and nitrification process itself in our oiled sites. But if you look at methane oxidizing bacteria, which are very similar in their structure to ammonium oxidizers and can actually carry out the process of nitrification, we actually see that in 2 of our 3 regions and overall, that methane oxidizing bacteria have positive correlations, and quite strong ones, with nitrification or unoiled sites. So you sort of have a double whammy here of the 2 sets of studies I've talked about, is that in unoiled sites, methane oxidizers are playing a big role in oxidizing methane, as their name indicates they should, but also at regulating nitrification rates. But as oil comes in, they are getting suppressed, and they are no longer oxidizing methane, so we have higher rates of methane flux. And we also see that they are not what is driving the nitrification rates. They're actually being driven more by the traditional ammonium oxidizing bacteria and archaea. We look within those nitrifying communities, and this is work that's led by Ann Bernhard. I'm not going to claim to fully understand every component of this. What I'm going to basically tell you is that you can actually identify individual components of that community. And so what these bars here are indicating, the different colors represent different clusters within the nitrifying community, and this is for ammonium oxidizing archaea and bacteria. And what you see here is that while, for example, in bacteria, you may see some differences between the bars for terrebone, western Barataria, and eastern Barataria. Within those regions, comparing oiled and unoiled, you do not see strong differences in at least who's present. But we do see some differences and shifts in terms of which of these different nitrifiers are responsible and have positive correlations with nitrification rates. Where in terrebone, we actually have several of these different groupings that have positive correlations in the unoiled sites. Whereas in western Barataria, we have some that actually have relationships with the oiled sites only, and then in eastern Barataria, we have some that have relationships with both. So we're seeing a shift in who is in these communities and who is responsible for carrying out these processes. So it may not be the predicted response of big changes in nitrification rates between oiled and unoiled status in 2012, but we are seeing more subtle changes in the communities that influence the relationships that those organisms have with carrying out these important processes. All right, so the last set of data I want to talk about is phosphorus sorption. We have highly variable rates of phosphorus sorption within our region, comparable to most of the range that's seen throughout most coastal wetland environments. But we do not see any differences between region or between oiled and unoiled sites. part of that is because while we have about an order of magnitude range across our whole data set, within each region we see about a 5-fold difference. So the range we see within a region and even within a site is often as big or nearly as big as we see across the entire data set. We do have very good predictive capabilities for what is driving phosphorus sorption. If you look across all marshes, 70% of the variance in phosphorus sorption is explained by iron. And actually within each of the regions, it's about 90%. And if you actually exclude one outlier here, this also is about 90%. But they're all individually greater than 50%. So one thing that phosphorus sorption allows you to do is to assess what people use in northern forests trying to talk about nitrogen saturation. We can do a similar thing looking at phosphorus saturation. And so what you're doing here is coming up with a theoretical estimate of how saturated your soils are for phosphorus. And so values that are above 0.15 would be saturated. You see that we only have a couple of sites or plots within Terrebonne that sort of exceed this eutrophication tipping point. We can also calculate a metric called soil phosphorus storage capacity, which says how much more phosphorus could be stored in the soil. And so we see here that these Terrebonne sites that are closer to saturation have less capacity to store future soil, at least future phosphorus loads, at least for some of the sites. But in general, we don't see a very big difference across our region. But when you look across the whole data set, as phosphorus sorption sites become less available, which is indicated by an increase in saturation ratio, phosphorus sorption index decreases exponentially, and as soil storage capacity for phosphorus increased, PSI also increased. So I mentioned that we have a lot of variability across or even within our sites in a lot of these biogeochemical processes. And this may be a little counterintuitive to people because when you go out and you look at our marshes, they don't look like they see in a textbook where you have a low marsh and you see this huge gradient in elevation. You look out, our marshes look really flat. But what we've noticed is that we see some interesting patterns with marsh position. So this is greenhouse gas flux, this is the net CO2 flux. Again, the open symbols are unoiled and the gray are oiled. And this is increasing distance from the marsh edge. So we see stronger patterns, both show an increase as you move into the marsh, and in the R squared in the unoiled is about 0.7, and it's highly significant. When you look at methane, your eye is drawn to this apparent decrease in the oiled sites, but in fact this small subtle increase in the unoiled sites is highly significant, has an R squared of 0.95. And this is only sort of marginally significant of a decrease. And N2O didn't show a spatial pattern with distance for oiled or unoiled marshes. We look in 2013 and 14, CO2 shows a stronger increase with distance in the unoiled sites than in the oiled sites, as we saw here in 2012. Methane trends for declines in oiled and increases in unoiled marshes. N2O. Unoiled marshes tended to increase with distance, and oiled marshes significantly decreased in 2013, but showed no pattern in 14. So again, N2O is not as clear of a pattern. This is greenhouse gases. We look at the nitrogen cycle. So now, not only do we have within each region, we have all three regions. Nitrification potential increases with distance in Terrebonne, decreases in Western Barataria, and shows no pattern in Eastern Barataria. We look at the ammonium oxidizing archaea and bacteria, we see the exact same pattern. Increase, decrease, no relationship with distance. If we look at total nitrate reduction and the near-S gene, which is the indication for denitrifiers, we see a similar pattern. Increase in Terrebonne, decrease in Western Barataria, and no pattern in Eastern Barataria. Look at the phosphorus sorption. We see increases in Terrebonne. While not significant, we see a trend towards a decrease in Western Barataria and no pattern in Eastern Barataria. So what's happened? So I mentioned these marshes look relatively flat, but these small scale changes in elevation have basically made us just look at those marsh elevation gradients in a micro scale. Because over the course of these 5 to 20 meters in terrebone marshes, we see that we have an increase in the order of 10 centimeters. In western Barataria, we actually decrease by about 8 centimeters. In eastern Barataria, we don't see a change. How does this translate to soil properties? Well, if you look at organic carbon across those 3 regions, we have an increase in terrebone, a decrease in western Barataria, and no pattern in eastern Barataria. And just to save us from all looking at hundreds of graphs, just looking at terrebone here and looking at the pattern increasing distance, we see organic, the water content, total nitrogen, total phosphorus, iron, aluminum, and plant available phosphate all increase with distance from the marsh edge. So, thought I had a summary bullet here. So we have these patterns that within our marsh that seem to be dictated by differences in elevation that change the inundation patterns and have corresponding changes in the soil properties that are driving a lot of our biogeochemistry, which we didn't realize before we started the study, which is something you need to consider when you're trying to figure out how to manage moving forward. So oil sites were lower in CO2 and N2O and higher in methane fluxes. We've seen this for about 4 years so far. Our small-scale measurements were not detecting the same level of response signal as we see in these whole system measures. Nitrification rates do not show consistent responses to oil. We saw no effects in 2012, but some differences in 2003 in at least terrebone. The ammonium oxidizing archaea and bacteria abundances do not show consistent oil responses. And no overall community differences in ammonium oxidizers, but there are differences in the correlations of the individual population with rates between oil and unoiled sites. And phosphorus sorption did not show a strong response to oil. But one of the real striking responses is that the process rates display high spatial variability within our marshes, and it's related to variability in soil properties, which appear to be, at least in part, regulated by differences in elevation and hydrology. And I want to end by just putting things in context that, it's difficult to detect impacts and recovery from oiling due to a variety of differences that we see in the timing, spatial distribution, extent of oiling, the overall and specific compound degradation rates, the loss of habitat as a result of oiling, decreasing amounts of residual oil across the coast as you move further and further away from the spill, and the re-oiling events that Dr. Turner mentioned earlier, like Hurricane Isaac, have added to the complexity. This is an area here that was sampled about 2 weeks after Isaac that did not look like this prior to Isaac. And the oil samples indicate that there was a stimulation in oil, and it was of a Macondo signature. And these marshes are very complex. There's a lot of different drivers that are being forced upon these systems in the way that they function. I think it's really important that when we make management decisions, we need to remember that these coastal ecosystems are faced with multiple stressors that not only are likely to influence the functioning of the systems, but also how they'll respond to future spills. Because as Dr. Twilley alluded to, trying to apply something that happens in one region to another region may be complicated. Even within your own region, if you're experiencing changes in your salinity regime, the degree to which you're inundated, how your vegetation changes, those things may all influence the way that your system functions and make it a little bit more difficult to detect or predict what the response to a future spill may be. And with that, I'll take any questions. Thank you.
So what do you think the long-term impacts of your findings are? Like, why should this audience care about nitrification or methane rates, methane fluxes? What's the implication?
So I've got 30 minutes to answer that question. Well, I mean, it's a very good question. I think one of the things that you're trying to evaluate we can never fully restore these systems. We're fighting a challenging battle as we continue to lose land, regardless of when oil spills happen. But if we understand more about the way that these systems function and how the roles that they play, you know, the wetlands serve an important location on the landscape where they're between the delivery of nutrients coming from the river that's draining the entire watershed and exporting offshore. So changes in the ability of that system to retain, and even more importantly perhaps remove nitrogen from that system before it goes offshore and impacts hypoxia is very important. So if you have a change in your system that influences the rate at which denitrification takes place, that's important to know, because that's not retained in the marsh. It's actually removed from the system or released to the atmosphere. Understanding what happens with, interested in how the relative rate of sea level rise in our region, one of the things that drives that is greenhouse gas fluxes. So if you have a change in your system that changes the relative contribution of methane to the atmosphere, that has long-term feedbacks that influence the rate of sea level rise. Because as you increase methane flux, you increase temperatures and decrease sea ice and increase water levels. So there are a lot of feedbacks here. But I think if we want to understand what components are the most important for us to be able to manage or make those decisions, we need to understand better the role that these systems really play. And so understanding how these things are connected, I think, is really important.
Would you like to tie these to the carbon credits for marsh restoration, these results?
Not until you incorporate the plants into the equation, which we're doing. I think we've got to, and that's always a.
There are claims or assertions about carbon flux credits. Right. And that's dependent upon CO2 flux and methane flux.
Right.
And so you can take one sample site versus another site and you get very different results.
Yes.
As you've shown.
Right, yeah. I guess I was misinterpreting how the context of what you're asking. But yes, these are things that need to be considered if you're going to be making decisions about carbon credits for trade-offs. In terms of some sites are going to have different valuations associated with them based upon what they mean for gas fluxes. So nobody asked the obvious question about why might there be differences in these. We see these strong spatial patterns across these three regions. You know, why might there be differences?
Why?
Well, anybody got any? Who's got ideas? That could be. I think part of it is so, you know, if you look at the way most of our marshes originally form, you end up, you know, outside the channel, you tend to have these sort of naturally high levees. And if you actually look at sort of healthy marshes that have not had as much erosion, you tend to have that levee that kind of comes down and it kind of rises up again. I pointed out that our Terrebonne sites, we had a couple sites that we've already lost 10 meters in 3 years. In many of those cases, what's happened is, and they also tend to be lower in absolute elevation, meaning they're inundated on a greater frequency than some of our other sites. They don't sit as high in the water. So my conjecture is that essentially our erosion rates have been so high at those particular sites that we've sort of eroded away those levees. And so you're beyond it. Because it's not, the erosion rate's faster than the resupply that's coming from those channelizations. That's my conjecture. Don't quote me on it, but that's one of the things that's there. There's clearly some big differences. And so this, you know, plays into, you know, these sites were selected primarily to go out there and see places where there was oil and nearby where there wasn't. But you get some different pictures in the way that these systems work depending upon, you know, which sites were selected. And so you need to think about this complexity when we're trying to evaluate comparing one site to the next.
Have you concluded anything different with the reaction of the marsh to the oil from the condo than previous oil spills?
No.
Say like a terrible parish. There's been a lot documented.
Well, there's been a lot documented, but none of the measurements that I showed you here are any of those. None of those measurements are made. So, I mean, yes, there are a lot of oil spills, and we've started to sort of look at some of those other spills that have happened and sort of see changes that we see in shoreline and things like that associated with it. But in terms of the biogeochemical processes and that functioning component of that system, there isn't data. I mean, I wish there was. Our job would be a lot easier if we actually had some baseline data on what these rates were. But it's a very good suggestion. We do have a wealth of oil spills around here, and we need to take advantage of that when we're trying to evaluate what we need to do moving forward. I think I'm going to be kicked off the stage, but I'll take another question.
All right.
Well, has there been a tracking of oil spills post-Monaco at these sample locations? As to whether or not there's other oil spills influencing these data sets?
Well, that's what Dr. Turner was alluding to before. I mean, and actually, because we have a different sampling regime than Dr. Turner does with his group, we have complementary oil samples that are collected. So, and frankly, I'm waiting on a lot of that data to sort of put some of these pieces together. But we can detect some of these things. For example, this picture right here, I mean, that we know was Macondo that happened after Isaac. We can tease apart, differences of, knowing whether or not, though, what I just want to say. So yes, we are addressing it, but one thing that is challenging is that heterogeneity that Dr. Turner alluded to, in the fact that if you go out to collect a sample that's 10 meters in, 1 month, then you come back the next month and you sample it, at 10 meters in, but you're a meter over, you may see some pretty drastic differences in those concentrations. So you may be low 1 month, the next month might be high. That doesn't mean that there actually has been a new oiling event. So we need to look at more specifically at those biomarkers to indicate, how degraded the age of that oil is, and we need to look at it over time. So you have a trend. You may have a low point and a high point, but overall you have some sort of trend or trajectory associated with it. So again, complexity is, I guess, the answer. And now I know I'm off the stage. So if you have questions, I'll be happy to take them later. Thank you.